All right, so today we are going to start the plant form and function, one of the most important lessons in the biology syllabus. You will get around maybe around 6 to 7 minimum amount of MCQs and also a definite essay and also a definite structured essay question. So, the beginning of this lesson is very smooth and very easy to understand. So what I would say you is now, when you're studying unit 4, like as I said, I have planned to complete within 5 weeks. Whatever the parts I'm completing today, before the next class, please finish revising and please completely memorize it. Don't keep it for later. That's what I say my microbiology students also when I told them recently, don't keep it for the next week to cram it. So today itself when I finish this, before next Wednesday you have to finish this off. Okay, so that is the plan. um, so let's get back to the lesson. Okay, when we talk about plant form and function, the first thing that we have to discuss is about the meristematic tissue. So before talking what are meristematic tissues, let's see what tissues are. What are tissues? Now, talking about a tissue is a group of one or more cell types which carries out a specialized function. So the cells combine together, cells get together to form a tissue. Okay. Now, the main type of tissue that we have to study here is the meristematic tissue. What are meristematic tissues? If they ask you the definition of meristematic tissue, you have to write what I have given here. It is an undifferentiated tissue, and it consists of cells which constantly divide under suitable conditions to produce new cells. This is the definition of meristematic cells. Basically, they are undifferentiated tissues which constantly divide, means it has the ability to divide under suitable conditions. Only if the environmental conditions are favorable, it will start dividing and it will produce new cells. Okay, in case if the environmental conditions are not favorable, meristems can undergo a dormant period, just like the seeds which undergo seed dormancy. Even these meristematic tissues, it can have dormant periods. Okay, so what are the characteristics of meristematic tissues? There are a few characteristics of meristematic tissues. They are living cells and they are isodiametric in shape. What is meant by isodiametric? Isodiametric isodiametric in the sense they are roughly spherical. Okay, almost like a spear, but not an exact spear, but almost like a spear. They are roughly spherical. And the third one, it has a central nucleus, it has a dense cytoplasm, and it has the ability to multiply. Also, another thing, it is both structurally and functionally undifferentiated. Okay. Now, in questions they ask you, I came across a question in my paper class last week. The question was, what is not a characteristics of meristematic tissue? And then they gave living, isodiametric, dense cytoplasm, ability to multiply, and having a central vacuole. So the answer is having a central vacuole, because there's no central vacuole present in the meristematic tissue. But what people do is when they just see the word central, they think, yeah, it is a characteristics of meristematic tissue. We came across that. No, please read the questions properly. It is the meristematic tissues has central nucleus, but it does not have a central vacuole. Keep that in mind. Now, in meristematic tissues, it has three overlapping zones. Okay, so the zones are, first of all, these meristematic tissues, they undergo cell division, and after that, consequently, they undergo cell elongation, and finally, they differentiate structurally and functionally to produce mature cells, and that happens in the zone of cell differentiation. So, what are the three overlapping zones? The zone of cell division, the zone of cell elongation, and zone of cell differentiation. Okay, then we have to study about different types of meristems. What are the different types of meristems? So there are three types of meristems that we got to study. First one is the apical meristem, second one is the intercalary meristem, and third one is the lateral meristem. So we'll see what these tissues do, what these meristematic tissues do. So the first one is apical meristem. So the apical meristem is important for the primary growth of the plant. What is meant by the primary growth? Primary growth in the sense increasing the height of the plant. Okay, the height can be increased by the lengthening of the root as well as lengthening of the shoot. So these apical meristems are present at the root tip and the shoot tip. Okay, the shoot apex or the root apex. So this apical meristem is important for the primary growth of the plant. Then comes intercalary meristem. So you can find intercalary meristems basically in monocots. Okay, you can find them in monocots like grasses. So these intercalary meristems are located in the bases of the stems, and leaves. For example, if you take a leaf like this, which is attached to the stem, at the base of the leaf and at the base of the stem, you can find intercalary meristems. So what is the use of having intercalary meristems? Intercalary meristems allows rapid regrowth of damaged leaves. So that is the function of intercalary meristems. Then we have lateral meristems. Okay, so lateral meristems, unlike the apical meristem, it is important in what? It is important in secondary growth of the plant. So secondary growth does not occur in all the plants. It occurs in all gymnosperms and most of the woody plants. Okay, so there are two types of lateral meristems as vascular cambium and coke cambium. So what is the function of vascular cambium? The vascular cambium, it will add the secondary xylem and the secondary phloem in the primary structure. Okay, to the primary structure, it will add the secondary xylem and the secondary phloem and produce the secondary structure of the stem and the roots. And then we have the coke cambium. So this coke cambium, it produces coke cells, and eventually these coke cells will lignify and become dead cells. So together, the coke cambium and the coke cells together, it will form the periderm. So periderm is a tough, thick covering that replaces the epidermis in the primary structure. Okay, so that is the function of the lateral meristem. So basically, lateral meristems are important in increasing the circumference. What is circumference? The diameter. So it is important in increasing the diameter of the plant or the circumference of the plant. Okay, did you understand? Do you have any doubts up to now? Okay. Then let's move on to the next section, that is the primary growth of the roots and the primary growth of the shoots. Okay. Now, before moving on to the next section, so here I have included some images. Here you can see the root apical meristem, and here you can see the intercalary meristem. See, at the bases of the stem, you can find intercalary meristem. So these are nodes. You can see these nodes very clearly in bamboo plant and all. You can find these nodes. Okay, it is present in the monocot stem. All right. Let's move on to the next section, that is the primary growth of the root. What is the primary growth of the root? Primary growth of the root is the elongation of the root due to the activity of the primary meristems that is located at the root apex. So if you consider the root, at the tip of the root, you have the root apical meristem. Root apical meristem. So we know that this root apical meristem, whatever the meristem it has the ability to produce new cells. So therefore, what happens is when the new cells are produced, it undergoes cell elongation and finally lengthen the root. So the basic idea is the primary growth of the root is the elongation of the root or the lengthening of the root due to the activity of primary meristems. What does that mean? Due to the new cells that are produced by the root apical meristem located at the root apex is what you call as the primary growth of the root. Okay, and then we have this is the primary, the apical meristem. The root apical meristem is here. So I'll explain you the process, what happens here. So this root apical meristem has the ability to produce cells outwards as well as inwards. Okay, it has the ability to produce cells outwards as well as inwards. So these cells which are produced outwards, it will form the root cap. It will form the root cap. So what is the function of this root cap? This root cap will prevent damaging the root apical meristem from friction when it goes through the soil. That means when this cell elongate, what happens is, if this is the soil and if this is the level of the root now, when this cell, when this root starts elongating, it will push through the soil. When it pushes through the soil, it will come across a lot of friction. So we have to protect the root apical meristem from this friction. So therefore, this root cap provides protection to this root apical meristem. And then the cells that are produced inwards, they will enter the next division, that is called the zone of cell elongation. So in the zone of cell elongation, whatever the new cells that are produced, they will elongate. Okay, they will increase their length. They will elongate usually 10 times the initial length of the cell. So when the cells start elongating in this region, the root will be pushed more and more into the soil. Okay, and after cell elongation, the cells will enter the zone of cell maturation or the zone of cell differentiation. So in this zone, what happens is the cells become structurally mature as well as it will become functionally mature. So this is the basic scenario of the primary growth of the root. Okay, now this is the primary structure of the root. So here you have the vascular cylinder, and then here you have root hairs. Root hairs are unicellular structures, and then we have what you call the root apical meristem here, and then we have the root cap here. So in case if they ask you an essay from this section, you need to draw this as well. All right, moving on to the next section, that is the primary growth of the shoot. So just like the primary growth of the root, what is meant by the primary growth of the shoot? Primary growth of the shoot in the sense the shoot starts lengthening due to the new cells that are produced by the primary meristems or the apical meristems that is located at the shoot apex. If I say you the correct definition, that is the elongation of the shoot due to the activity of the primary meristems located at the shoot apex. Now, if you consider the diagram that I have drawn here, this is how the shoot apex will appear. Okay, the shoot apical meristem is a dome-shaped mass. Dome-shaped in the sense the shape is like this. Okay, it is a dome-shaped mass of dividing cells. So this part is the shoot apical meristem, and this shoot apical meristem is protected by two leaf primordia on either side. We call them as leaf primordia. Okay, and here also, here what happens is the shoot apical meristem, it will produce cells only inwards. Only inwards. It does not produce cells outwards. So the cells that are produced inwards then will undergo elongation. So when it undergoes elongation, what happens is the stem will start lengthening. And after it undergoes elongation, it will structurally and functionally mature. So this is the basic process happening in the primary growth of the shoot. So the shoot apical meristem, it is protected by what? It is protected by the leaf primordia. So this is the diagram. Here you have the shoot apical meristem, and then it is protected by the leaf primordia like this. And then at this section you have auxiliary bud meristems. You need to know this. You need to know how to draw this and mark this. You have auxiliary bud meristems like this, and then these are young leaves. Okay, these are young leaves that are developing on either side. And then these small tissues, okay, these small tissues that appear inside this, that is not visible. So within the leaf primordia, you can see another tissue system that is going on like this. So that is the developing vascular tissue, which is the xylem and the phloem. Okay, so now we discussed about the primary growth of the root and the primary growth of the shoot. So we have to know the differences. Sometimes they will ask in a structural essay question to compare between the shoot apex and the root apex. Okay, so we obviously know when we consider the shoot apex, it is found at the tip of the shoot, and the root apex is found at the tip of the root. Then comes the protection. The shoot apex is protected by what? It is protected by the leaf primordia, and the root apex is protected by the root cap. Then comes the way it produces cells. As I said to you, the shoot apex, it produces cells only inwards, but root apex produces cells both outwards and inwards. So the cells produced outwards, they will form the root cap, and the cells produced inwards, it is responsible for the elongation of the root. Did you understand up to this point? Yes, clear. Okay, so the next thing is that we have to talk about the tissue system. So when we talk about the plant tissue systems, the plant is composed of three types of tissue systems. The first one is the dermal tissue system, second one, the ground tissue system, and finally we have the vascular tissue system. So first of all, we'll talk about the dermal tissue system. Dermal tissue system is the outer covering of the plant. Okay, it is the outer covering of the plant. All right. Now, the example for the dermal tissue is the epidermis. Okay, example for the dermal tissue is the epidermis. Now, let's see what are the features of this epidermis. So, the features of this epidermis include that is the outermost protective covering. Okay, that is the, like, the outer limit, that is the barrier, that is the outermost protective covering. And the epidermis, it is a single cell layer. Okay, epidermis is a single cell layer, and the cells are tightly packed to each other like this, just like the epithelial tissue in humans. Okay, it is a single cell layer and it is tightly packed to each other like this. And within this epidermis, you can find several specialized cells. So these special cells include the guard cells, trichomes, and the root hairs. So the root hairs are unicellular structures. So first of all, we'll talk about the guard cells. Guard cells are the only type of epidermal cells which contains chloroplast. Okay, that is the only epidermal cell which contains chloroplast. And guard cells are responsible for gaseous exchange. Then we have trichomes. When we talk about trichomes, trichomes can be two different types. The first one is you can have hair-like trichomes, and the second one is you can have gland-like trichomes. So first of all, if we talk about the hair-like trichomes, the hair-like trichomes will reduce water loss. That is the first function. It will reduce water loss. And number two, the shiny hairs, it has the ability to reflect excess light. It reflects excess light because when the plant is exposed to excess light, um, the chlorophyll and the other photosynthetic pigments can be damaged. So therefore, this hair-like pigment, hair-like trichomes, which has a shiny nature, it will reflect the excess light. Then we have gland-like trichomes. Why do I call it gland-like trichomes? The reason is it will secrete chemicals. Okay, it will secrete chemicals, and these chemicals provide defense against insects, pathogens, and herbivores kind of organisms. Okay, and then we have root hairs. So root hairs are unicellular structures, and root hairs are important in the absorption of water and mineral ions from the soil solution. So now, by discussing about the features of these three types of cells, I already mentioned two functions of the epidermis as well. And apart from that, what are the other functions of the epidermis? The first one is it provides defense against physical damage and pathogens. Basically, it protects the plant against physical damage and pathogens. And apart from that, the cuticle that is present above the epidermis. So here, if you consider this image that I have given here, you can notice that above the epidermis there is a cuticle. So this cuticle, it will prevent water loss. That cuticle is important to prevent water loss. All right, so that's all about the epidermis and the dermal tissue system. Then we have to move into the ground tissue system. So, talking about the ground tissue, mainly if you consider a certain plant, the ground tissue consists of two parts. The first one is the cortex and the pith. Now, if you consider just the structure of a stem, now suppose this is the structure of the dicot stem, and then the vascular bundles will be arranged as a ring like this. The gap that is between the vascular bundles and the epidermis, this section is a ground tissue, and that ground tissue we call it the cortex. And then the ground tissue that is found between the vascular bundles, or else at the center of the vascular bundle, we call it the pith. Okay, so the ground tissue is mainly composed of the cortex and the pith. In common, the specialized function, the ground tissues have specialized functions. Number one is storage. Number two, it can carry out photosynthesis. Number three, it provides support. And finally, it can involve in short distance transport. So these are the common functions of all the ground tissue that we are going to discuss next. So we'll be discussing about parenchyma cells, collenchyma cells, and sclerenchyma cells. So all these in common have these four functions known as storage, photosynthesis, support, and short distance transport. Okay, then we have parenchyma cells. So talking about parenchyma cells, they are living cells even at functional maturity. Even if they are functionally mature, they are living cells. So talking about their structure first of all, so this is how parenchyma cells will look like. Here you have the nucleus and there is a large central vacuole. Okay, there is a large central vacuole. These parts which appear in white, those are large central vacuoles. And if we talk about the cell wall of parenchyma, the cell wall of parenchyma is relatively thin and it is flexible. Okay, and parenchyma cells, most of them, they do not have secondary cell walls. So what are the features of parenchyma cells? They are relatively thin. They have a relatively thin and a flexible cell wall. They have a large central vacuole and they are living even at the functional maturity. So those are the features of the parenchyma cells. Then comes the functions. What are the functions of the parenchyma cells? Talking about the functions, number one, it performs most of the metabolic functions of the body. Example, synthesis of various products. Okay, basically photosynthesis. The parenchyma cells, it can carry out photosynthesis, and photosynthesis is where food is produced within the plant. So in order to synthesize various organic products, we need parenchyma cells. So it performs most of the metabolic functions of the body. And number two, it can involve in storage. So where can you see the storage function of the parenchyma cells? So there are some cells in the roots and the stems which contain a structure called the plastids. And these plastids have the ability to store starch. Okay, that is number two. And number three, these parenchyma cells, even after they functionally mature, still they will have the ability to divide and differentiate under suitable conditions. Okay, still they have the ability to divide and differentiate under suitable conditions. And this property is very important in wound repair, and also due to the ability of multiplying by dividing and differentiating, we can produce a large number of plants by using a single parenchyma cell. So this is important in tissue culture practices. We already learned about tissue culture in Unit 10, all right? So this is important in tissue culture practices. So those are the functions of the parenchyma cells. Then we have the collenchyma cells. So talking about the collenchyma cells, here I have given you the longitudinal section. When you view it longitudinally, how it appears. I wanted to show it because collenchyma cells, one of the function is they are generally elongated. By looking at the cross section or the transverse section, you will see it is not elongated, no Kiela. But if you view in the longitudinal section, you can see these cells are generally, they are elongated. And if we talk about the cell wall of the collenchyma cell, the cell wall is thicker than that of the parenchyma. The primary cell walls are thicker than that of the parenchyma cells. And if you look at the corners of these cell walls, they are unevenly thickened. The corners are thickened. So we can say the primary walls are unevenly thickened as well. Okay. And what are the other features of the collenchyma cells? These cells are also, they are living, okay, at functional maturity, they are living, and also they are flexible. They are living and flexible. Usually, you can find collenchyma strands in young stems and petioles. Petioles are the basic, the base of the stem of a leaf. If you take the leaf like this part is the petiole. So you can find collenchyma strands beneath the epidermis of these young stems and petioles. So basically, since it is living and flexible, when the young stems grow and when the petioles grow along with the young stems and petiole, even the collenchyma cells will also grow. So what is the function of collenchyma cell? The function of the collenchyma cell is that it will give mechanical support. It will provide mechanical support to the leaves and the stems without restraining their growth. Without restraining their growth in the sense, just because collenchyma is present, the cells don't become dead. Okay, without any hassle, the cells have the ability to grow by themselves, even the collenchyma is present. So the function is it gives mechanical support to the leaves and the stems without restraining their growth. Is it clear to you? Any doubts so far? Understood. Good. Next, we have to talk about the sclerenchyma cells. So, when we talk about sclerenchyma cells, the sclerenchyma cells have secondary cell walls. Okay, the sclerenchyma cells, they have secondary cell walls, and these secondary cell walls, they are thickened by large amount of lignin. You just can't write it is thickened by lignin. Okay, just not only a little lignin is present in sclerenchyma cell walls. Okay, so the secondary cell walls have large amount of lignin, and therefore due to the presence of large amount of lignin at maturity, they are considered to be dead cells. And the sclerenchyma cells are of two types. The first one is sclerates and the second one is fibers. First, if we talk about sclerates, they are short and wider. So here you can see a sclerate. They are shorter, they are shorter, they are wider, and they do not have a particular shape. So they have an irregular shape. Okay, we can't define a shape for the sclerates. So um, these sclerates, they have very thick lignified secondary cell wall, and you can find sclerates in places where growth has stopped because because the sclerenchyma cells, they are dead. Okay, if they are present in places where the growth occurs, they cannot grow with the plant or the base of the stem that they are supporting. So therefore, the sclerenchyma cells, the sclerates and the fibers are present in places where the primary growth has already ceased, so the growth has stopped. For example, you can find in nutshell, seed coat, and the flesh of coarse fruit. Flesh of coarse fruit in the sense like hard fruits, like pears. Pears is one of the best example for hard fruits, and then we have guava. So these are coarse fruits. Okay. And then we have fibers. Now, fibers compared to the sclerates, they are usually elongated. Okay, they are usually elongated strands. And if we talk about their functions, we can say they are long, they are slender. Slender in the sense they are thin. Okay, they are very thin, and they have tapered ends. Tapered end in the sense this end, the pointed kind of end is what you call as a tapered end. So we can use fibers, okay, the sclerenchyma fibers to commercially obtain fibers like coconut husk fiber, hemp fiber. Okay, fibers are again we can use sclerenchyma cell. So what is the function of the sclerenchyma cell? The function is it provides support and strength. Okay, it provides support and strength. That is the function of sclerenchyma cells. All right. Next, we have to talk about the vascular tissues. So after we talk about the vascular tissues, okay, the phloem and the xylem, then we have to talk about the primary growth or the primary structure of the root and the primary structure of the plant stem. So that's it, like that. Those are the sections that we have to discuss today, but still I will give you an essay to write before we move into the vascular tissues. Okay, let me check for the essay. Give me 2 minutes. I hope You can write. You said you have a leg injury. How are you? Little bit. What happened to you? Okay, okay. Hope you're fine now. Get rest. So your essay is going to be briefly describe the structure and function of the ground tissues of the plant. Of the ground tissues of the plant. I'll be providing the marking scheme. So before that, I'll give you around, now it's 4:46. I'll give you time till 5:10, which is around 25 minutes. Um, if you want, you can refer the book for 5 minutes and take time till 5:15. Please write an essay. Okay. I wouldn't mind even if you switch on your video, if it's possible for you. I saw you anyways. Your video is switched on. So you will get 25 minutes for this. Now it's 4:46 maximum. When you're done, leave me a message and then we can move on to the vascular tissues and the primary structures of the root and the stem. Okay. So our essay was to write about the ground tissues. Now, when you write about the ground tissues, I'll share you the marking scheme. So you have to say the ground tissue consists of the main types of cells called the parenchyma cells, collenchyma, and the sclerenchyma. And then comes the parenchyma. Then you have to tell about the primary cell wall of the parenchyma, and then tell that it is living, and then it is thin, and then It has a large central vacuole, and then you also have to describe about the functions now. So you can describe about the functions separately, or else you can describe along with this as well. Okay. And apart from that, you have to draw the diagram and name it completely, label it entirely, then only you will get the few complete marks. And then you move on to the colon cells. You have to mention it is generally elongated and it has primary cell walls which is thicker than that of the cell walls of the parenchyma cells. And then you can mention the places where you can find these colon cells, and then you can mention the other features, mention the function, and then draw the diagram and get back to sclerenchyma cells. So likewise, you have to write it very neatly, and you don't have to you should not write it in point form. You should always write it in the form of paragraphs, as I always said to you. Write in separate paragraphs. Don't write everything together. All right. Now I'll share you the marking scheme. Let me share it with you now. Okay. So after the class, please mark your essay using the marking scheme, and then let me know your marks. Okay. Right. Then we have to talk about the vascular tissues. So talking about the vascular tissues, there are two types of vascular tissues. The first one is the silent tissue, and the second one is the phloem tissue. So first of all, we'll talk about the xylem tissue. So what is the function of the xylem tissue? The function of the xylem tissue is to transport water and minerals. Now, this xylem tissue is composed of four other cells called the xylem vessel element, tracheids, xylem fibers, and xylem parenchyma. So overall, in general, if we discuss the functions of these specific cells, xylem vessel elements and tracheids, they are involved in conduction of water. They conduct water mainly. That is their main function. And then if we talk about the fibers provide strength. Always remember, fibers are a type of sclerenchyma. So they are dead at functional maturity. So always they provide mechanical strength. Then we have the parenchyma. The function of the parenchyma is very important. The xylem parenchyma involves in storage as well as in radial transportation. We'll be studying about the radial transportation maybe in our third or fourth classes, probably in the third class. So radial transportation is where the water and minerals that are uptaken by the root hairs are transported into the xylem of the root via the cortex. Okay, so this radial transportation occurs in three different parts: the apoplastic part, symplastic part, and the transmembrane route. Okay, so xylem parenchyma is involved in this radial transportation. Then we have to study about the xylem vessel elements and the tracheids in detail. So we'll check the specific features of these cells. So in this diagram, you can see the xylem vessel element and the tracheids. So first of all, we'll talk about the vessel elements. These xylem vessel elements, they are not present in all plants. Okay, they are present in all the angiosperms. It is present in all the angiosperms. What are angiosperms? Angiosperms are the flowering plants. At the same time, it is present in some gymnosperms. Okay, angiosperms and some gymnosperms. So what are the some gymnosperms that vessel elements are present? So gymnosperms, we can discuss three phyla under gymnosperms. So not all three phyla has xylem vessel elements. Only phylum Neophyta has vessel elements in it. Okay. Then, when we talk about the features of this xylem vessel element, it is cylindrical. So you can see it is cylindrical and it is a long structure. And when you compare with the tracheids, it is wide. Okay, it is wide as well as compared to the tracheids. Now, in this diagram, it is not obviously visible. Usually, tracheids are longer compared to the vessel elements. So the vessel elements are wider, shorter, and they have thinner walls than tracheids. So the tracheids have thicker walls compared to that of the xylem vessel elements. Okay. Now the xylem vessel elements are also dead cells. Now, when it comes to the xylem tissue, there are three dead cells, and one cell which is living. The xylem parenchyma is the only cell which is living. All the other three vessel elements, tracheids, and fibers, they are dead at functional maturity. So, since these vessels are also dead at functional maturity, their walls are thickened by lignin. Okay. So, what is the purpose of the thickening of the wall by lignin? That is to prevent collapsing of the wall under the tension of water transport, because water transport occurs continuously and there will be certain tension that is felt by these vessel elements. In order to prevent collapsing of the vessel walls, lignin provides strength to this vessel walls. Okay, and then if you see the xylem vessel elements, you can see these end regions. These end regions, they have perforation plates. Perforation plates in the sense these plates have pits like this. Okay, they are perforated, perforation plates, and you can see these walls, the lateral walls, they have small openings or apertures like this. So these small openings or apertures, they are called pits. Okay, and on the at the end of the vessel elements, the plate kind of a thing that you have, or else that what do you call the perforated lid. Okay, the perforated lid, that lid you have, it'll like it'll provide the environment for the continuous conduction of water. So these are called perforated plates. Perforated plates. So what happens is, how does the xylem tissue forms? The xylem tissue is formed by the end-to-end connections of the perforation plates of each vessel by like this. Okay. So each xylem vessel elements will connect end to end at its perforation plates. So through these perforation plates, the water can move freely inside. Okay, that's all about the xylem vessel elements. Then we have the tracheids. So tracheids, unlike the xylem vessel elements, tracheids are found in all vascular plants. It is present in all vascular plants. And these are long thin cells, and they have tapering ends. So I said to you what tapering ends are. Tapering ends are these, corner, like, the pointed shaped corner is what you call as a tapering end. So these tracheids, they have long thin cells with tapering ends. Similar to vessel elements, tracheids also have walls that are thickened by lignin, and these walls are interrupted by pits, just like in xylem vessel elements. Okay, and how does water move in tracheids? Water move in tracheids through these pits. From end to end through the pits, water movement occurs. So the purpose of having lignin here is also to prevent the collapsing of the tracheid wall against the tension of water transport. So that's all about the vessel elements and tracheids. Do you have any doubts? Is it clear to you? Do you have any doubts, and is it clear to you? Yes, understood. Clear. Right. Next one is the phloem tissue. So talking about the phloem tissue has four cells once again: the phloem parenchyma, the sieve tube element, companion cell, and the phloem fibers. So phloem tissue is considered as a living tissue because except the fibers, all other cells are living. Okay. Now, you can find all these four cells: the sieve tube element, companion cell, the phloem parenchyma, phloem fibers, all these four are present only in angiosperms. Okay, only in angiosperms. But when it comes to the seedless vascular plants are categorized as seedless vascular plants and seeded vascular plants. So when you consider these seedless vascular plants and the gymnosperms, once again, what are they? The seedless vascular plants and gymnosperms, they do not have all these four cells. In the place of sieve tube element and companion cell, it has another long narrow cell which is called the sieve cell. So you can find sieve tube element and companion cell only in angiosperm. So in other seedless vascular plants and gymnosperms. In the place of sieve tube element and companion cells, you have long narrow cells which are called the sieve cells. Okay, right. Then let's talk about this phloem tissue. When we talk about the phloem tissue, mainly we are talking about the sieve tube element and the companion cell. So this is the sieve tube element here. Okay, this is the sieve tube element here. So there are certain features of the sieve tube element. So what is the function of phloem tissue? The translocation of food. Okay, whatever the products of photosynthesis that are transported via the sieve tube element. So in order to do this transportation very freely, these sieve tube elements should have several modifications. The modifications are they lack a nucleus, ribosome, distinct vacuole, and cytoskeletal elements. Okay, so the sieve tube element, they don't have cytoskeletal elements, they don't have a nucleus, they don't have ribosomes, and they don't have a vacuole. At the same time, the cytoplasm, the cytoplasm of these sieve cells, they are reduced to the periphery. Okay, that means cytoplasm is not present entirely like this. You can find cytoplasm only at the corners of the sieve tube element. That is because to allow the movement of the nutrients more freely. So the adaptations are cytoplasm is reduced to a peripheral layer, and also it doesn't have nucleus, ribosomes, vacuole, and cytoskeletal elements. Okay. Now, there are just like the xylem vessels, the sieve tube elements will align one on top of the other, and at the end of each sieve tube element, you can find a sieve plate. Okay, sieve plate. These sieve plates, similar to the perforated plate that we found in the xylem vessel element, this also consists of pores like this. Okay, this is a porous plate. So this sieve plate, it allows the movement of the fluid from one sieve tube element to the other. Okay, so this is a porous plate here, this is another porous plate here. So the sieve plates will allow the movement of the nutrients from one sieve tube element to the next sieve tube element. So that's all about the sieve tube elements. Then we have to talk about the companion cells. Now, companion cells, they do not conduct. Okay, they do not conduct nutrients. So therefore, companion cells are considered as non-conducting cells. So these companion cells are found along with the sieve tube element. Can you see? They are like together. Okay, so they are joined together by numerous plasmodesmata. Plus, I hope you can remember what plasmodesmata is. We studied in unit 2. Okay, so these companion cells are always found alongside with the sieve tube element, and this connection between the sieve tube element and the companion cell is provided by numerous plasmodesmata. Okay, and then this companion cell, unlike the sieve tube element, it has a nucleus, and then it has ribosomes, and then it has cytoplasm. It has all the characteristics just like a normal cell. So whatever the processes that is occurring in the sieve tube element is governed by or controlled by the nucleus and the ribosomes of the companion cell. So the function of the companion cell, or the use of the companion cell, is that it will help in phloem loading and other organs unloading. What is phloem loading? Phloem loading in the sense, if you take the phloem tissue like this, usually the sugar source produces the what do you call the nutrients. So from the source, the transportation of the nutrients into the sieve tube element is what you call as phloem loading. And then at the sink, the phloem, the nutrients that are transported via the phloem tissue will be unloaded. That is known as phloem unloading. So companion cell is important for phloem loading and unloading. Did you understand? Is it clear to you? Yes, okay. Next, we have to talk about the plant growth. The plant growth, basically the primary structure of the stem and the root. So when we talk about the plant growth, what is growth? Growth is the irreversible increase of the dry mass, and it is associated with development. Okay, now when we consider the plant growth and the human growth is determinated. Determinated in the sense, after a certain age, after a certain period, we stop growing. But plants are not like that. Plants have indeterminate growth. Okay, plants have indeterminate growth. So therefore, they continue to grow throughout their lifetime. Give me a minute. One of the students is calling. So plants grow throughout their lifetime. So we have to talk about the primary structure of the root now. So when we talk about the primary structure of the root, we'll be studying about the dicot root and the monocot root separately. Hello? Yeah. Today we don't have classes. Walaikum salam. No. We have the class usually on Saturday. Yeah? Saturday 8 to 11. Ah, okay. Okay. You have I saw your message that you said you have completed the paper. Keep it with you. We'll be discussing eight structure diseases this week. Saturday 8 to 11. Yeah. No problem. Okay, no problem. So yeah, okay. So we have to talk about the primary structure of the root. Right. Let me share the resource book for that because I thought if you share the resource book image itself, it will be better because that is the image that you have to be familiar with. Okay. A minute. Today at what time do you have class? Ashif says. 6:30. Oh, I'll finish soon. Huh? All right. I hope you can see the screen. Now, when we talk about the primary structure of the root, okay, this is the dicot root and this is the monocot root. The main difference between the dicot root and the monocot root is the distribution of this vascular tissue. Okay, that is the main difference. Other than that, almost all the characters are same. Okay, there are a few differences. We'll learn what is that. Now, first of all, the outermost cell layer, that is the epidermis in both. You can see that. Okay. So what is the function of the epidermis? We already studied now. Epidermis also has specialized cells called root hairs because root hairs are present only in the roots because that is necessary for the absorption of water and minerals from the soil solution. So epidermis, it protects the inner parts of the plant. That is the main function of the epidermis. Apart from that, the root hairs that are present on the epidermis, that is important in the absorption of water and the minerals. Okay. Then between the epidermis and the vascular cylinder, you have the ground tissue layer. So this ground tissue layer is mainly made out of parenchyma cells. Parenchyma cells. And a special feature is this cortex that is found between the epidermis and the vascular tissue. It consists of the parenchyma cells and it has intercellular spaces, parenchyma cells with intercellular spaces. So what is the main function of the cortex? The function of the cortex is that it mainly stores carbohydrates because parenchyma cells, we studied under that. One of the main function is it stores starch. Okay, that is a carbohydrate. And the other functions are it transports water and minerals towards the endodermis. That means from the epidermis, from the root hair, when the from the soil when it absorbs water and minerals, that should be transported into the vascular cylinder, the xylem of the root. So that is done via this cortex. This is what you call as radial transportation. So radial transportation is also carried out by the cortex. Okay. Now, the innermost layer of the cortex forms a special structure that is called the endodermis. Okay, so always remember endodermis is the innermost layer of the cortex and therefore it is a single cell layer. It is a single cell layer. All right. So the feature, the main feature of the endodermis is that it has a special substance called the suberin, which is deposited in its cell wall. So this suberin, it is impermeable to water. So this suberin is deposited on the wall of the endodermal cells, and it forms something called the Casparian strip. It forms a strip kind of a structure, which is called the Casparian strip. So it will prevent the cortical apoplast from the vascular apoplast. That means I said you radial transportation has several pathways. One is the apoplastic route. So apoplastic route is where water and minerals are transported via cell walls and within the interior of the dead cells. Then what happens is, it will be transported via the cell walls of the cortex, and it can't move through the endodermis and get into the vascular cylinder. Why? Because the endodermal walls, they have suberin and it is impermeable to water. So therefore, the cortical apoplast is blocked by vascular apoplast due to the presence of a Casparian strip made out of suberin on the endodermal walls. Okay. And interior to the epidermis, interior to the endodermis, you have a structure called the pericycle. And this pericycle is not a single cell layer. It is made out of at least 2 or 3 cell layers. And these cells are parenchyma cell layers. Okay, parenchyma cell layers. Now, this pericycle in monocot root and dicot root is different. Even though the pericycle of monocot root and the dicot root, even both of them are made out of parenchyma and 2 or 3 cell layers, the pericycle of the dicot root only, it has a meristematic function. Meristematic function in the sense it has the ability to divide. But the pericycle of the monocot root does not have the meristematic function. So, this pericycle of the meristematic root, the pericycle of the dicot root, it is involved in the secondary growth. We will be studying how it is involved in the secondary growth under the secondary growth part. Okay. So the pericycle of the dicot root has a meristematic function. So therefore, it involves in the formation of lateral roots, and thereby it contributes to the secondary growth of the plant. Always remember, monocots do not undergo secondary growth. Only dicots undergo secondary growth. Therefore, pericycle of the pericycle of the dicot root, it has a meristematic function, and it involves in lateral root formation and involves in secondary growth. Okay. And inner to the pericycle, you have the vascular tissue. So just check the vascular tissue of the dicot plant. In the dicot plant, you can find the xylem as a star shape. Okay, the xylem is present as a star shape, and the phloem is present, phloem tissue is present in the grooves of the arms of xylem like this. But in a monocot root, within the vascular cylinder, there is a central core of parenchyma cells like this. Do they have any central core of parenchyma cells in dicot? No. There is a dense star-shaped xylem, and within the arms of the xylem, you can find phloem. So when it comes to the monocot root, at the center of the monocot root, there is a central core of parenchyma cells, and surrounding this parenchyma cells, there is xylem and phloem alternating like this. And also, there is another difference. The other difference is if you consider the dicot root in between this xylem and the phloem. Now xylem is like this, phloem is like this. No, I'll draw with a small gap. Okay, so in between the xylem and phloem, you can find the vascular cambium. So once again, that is important for the secondary growth. But do monocots undergo secondary growth? No. So therefore, they do not have vascular cambium. So that is the other difference. Did you understand the primary structure of the root, the dicot and the monocot root? Did you spot the differences clearly? Do you have any doubts? Yeah, clear. Okay. So we'll move on to the next section, which is the final section that we are going to discuss. That is the primary structure of the dicot plant stem and the dicot plant root. So when we talk about the dicot plant stem and the dicot plant root, there are several differences according to the arrangement of the vascular bundle and also according to the ground tissue and also regarding this vascular cambium and stuffs. Other than that, most of them are similar. Okay, first we'll talk about the dicotyledonous plant stem. So, similar to the roots, the outermost layer is what? The outermost layer is the epidermis. Let me zoom it in. Okay. So the outermost layer is the epidermis. What is the function of the epidermis? Now, unlike the root is found under the ground. The plant is exposed. The stem is exposed. It is an aerial part. So therefore, it is subjected to sunlight. It is undergoing heat and, it is exposed to the direct sunlight. So therefore, it can undergo dehydration, or we can also call it desiccation. So this epidermis is important to prevent desiccation and also to protect the plant from infections. So interior to the epidermis, you have the ground tissue. This ground tissue is made out of mainly parenchyma cells. Mainly parenchyma cells. And the ground tissue is differentiated into cortex and the pith. So, the ground tissue that is found outside the vascular bundle, this area, the ground tissue that is found outside the vascular bundle, this area, it is what you call it as the cortex. And the ground tissue that is found interior to the vascular bundle, this is what you call it as the pith. Okay. And this region which connects the pith and the cortex, you just write it as the ground tissue that is connecting the pith to the cortex. You don't have a special name to you have a special name to introduce that. All right. And after that, apart from the parenchyma cells that is found in the ground tissue, there are also collenchyma cells just beneath this epidermis. Now, this is the epidermis. Now, just below that, just below the epidermis, you have collenchyma cells as well. So, these collenchyma cells are present to provide strength. Apart from that, the cortex may also have some sclerenchyma fibers. Sclerenchyma fibers. So that is to provide additional support. That is to provide additional support. So in the ground tissue, you can find parenchyma. Apart from that, you can also have collenchyma just beneath the epidermis. Also, sometimes you can have sclerenchyma such as fibers in order to provide additional support. Then comes the vascular bundles. So these vascular bundles, they are arranged as a ring like this. It is very orderly arranged. And interior, the exterior section, that is the phloem, okay, the primary phloem tissue. And then the interior part, the vascular tissue which is facing the pit is what you call it as the xylem. And because this is a dicot plant, in between the xylem and the phloem, you have the vascular cambium, which is important in secondary growth. And apart from that, these vascular bundles, now this is a set of vascular bundles. So this vascular bundle is protected by a cluster of parenchyma cells. Okay, sorry, sclerenchyma cells. So this vascular bundle, if the vascular bundle is located like this, surrounding this, it can have a cluster of sclerenchyma cells in order to provide support and protection. Okay, so that's it regarding the dicot plant stem. Then comes the monocot plant stem. Monocot plant stem is easy because the monocot plant stem, the outer limit is the epidermis. So just similar to the dicot stem, epidermis will prevent desiccation and also it will protect from infections. And interior to the epidermis, you can find what? You can find the ground tissue. Okay. So this ground tissue is mostly made out of parenchyma cells. But here the ground tissue, it is not differentiated. Okay, but when it comes to the dicot stem, the ground tissue is differentiated into cortex and the pith. But in the monocot stem, the ground tissue is not differentiated into cortex and the pith. And then you have vascular bundles, and there's no particular arrangement for the vascular bundles. The vascular bundles are just scattered throughout the ground tissue. It is just scattered throughout the ground tissue. And another difference is there's no vascular cambium present in monocot plants. There's no vascular cambium. Okay. And And apart from that, even these vascular bundles that are present in the monocot roots are also protected by sclerenchyma clusters. Okay, so that's all you need to know regarding the monocotyledonous stem. But you need to know how to draw this, label this, and all the points that I mentioned under all these sections. Is it clear to you? Do you have any doubts? Yeah, clearly understood. Okay. So that's all we're going to do today. So for next week, we'll be starting off with the secondary growth, and then we'll be continuing probably until the water potential. So I have shared you the marking scheme. Please mark your essay before you go for your chemistry class and let me know your marks. And other than that, that's it. Then thank you so much for joining. I'll end the meeting. Okay.